Hi, my name is Kiara and I'm a PhD student in biomedical engineering at Brown University. And I'm sitting here today to present my poster titled Parallel DNA Extraction from Whole Blood for Rapid Sample Analysis in Genetic Epidemiological Studies. So genetic epidemiological studies require multiple patient samples that DNA will be extracted from. And as you can imagine, doing this for multiple patient samples requires a lot of time, cost, reagents, and equipment. So I created this microfluidic chip to reduce the burden of all of those. So how this microfluidic chip works is that it takes a 4 microliter sample of blood and lyses it, which means that the cells are being broken open and the DNA is being released, and then adds magnetic beads and a binding buffer to that, so that the DNA will bind to the magnetic beads. And from there, you place a magnet beneath the microfluidic chip and move it through a channel that contains a wash buffer, so the magnetic beads are being washed, and then they end up in an elution well which contains the elution buffer to elute the DNA off of the beads, which is done at a higher temperature of 55 degrees Celsius. So this microfluidic chip is about the size of my palm and has 6 of these reactions happening at once. So you can run multiple samples at once, so that significantly cuts down on your time. So to first validate the system, I compared this on-chip protocol to my off-chip, which is your manual protocol, and the off-chip or manual protocol requires 250 microliters of blood and 4 wash steps, and my on-chip protocol requires 4 microliters of blood and 1 wash step. So I compared these two and found that they had similar yield. And then also, when I was doing these experiments, I also found that with the microfluidic chip, the magnetic beads would clog it, and that was due to the large DNA content. So to reduce that effect, I started to dilute the blood in PBS, and that was done to reduce the amount of starting DNA that is in a sample. So what I found was that with a higher dilution factor, that more DNA was being eluted or recovered at the end, and that was about 4 nanograms is what I was getting. And lastly, I validated this further with testing the purity or the readiness of the sample for next generation sequencing. So first I did quantitative PCR of the Y gene, or of a gene found on the Y chromosome, and got similar results across the off-chip and the on-chip protocols. And then I prepared the DNA sample for sequencing and got a bioanalyzer trace, which was ideal for sequencing. So overall, this protocol can be performed in 40 minutes, and you can get 6 DNA samples out of it at once. And this microfluidic chip is able to prepare DNA for sequencing from a whole blood sample. So for future directions, I plan to actually deploy this chip in the field and try it to work with a clinician on a genetic epidemiological study, as well as apply this system to RNA, which could be very useful, especially right now. And lastly, integrate a dry blood spot system to extract DNA from those on a microfluidic chip. So that is my research, and I want to thank you all for listening, and I hope you learn a lot from my poster.